In this video, we will discuss about this co-indexing and transcriptome epitopes. This is one of the recent strategies by which we can do single cell RNA sequencing and find out what are the changes that are happened within the single cells. And this is some of the recent advancement in the field of biology, where we can identify the changes in the single cells. Previously, we are identifying the we can identify the changes in the group of the cells. However, with this sequencing, we can identify the changes in the single cells mRNA with the help of this transcriptome, that will read the mRNA within the single cells. So let us understand how this thing works. So it has this antibody derived tag, and how this antibody derived tag, what are the components of this antibody derived tags, is that it has this antibody that is specific to the cell surface protein. So, or we can say that it is against the cell surface protein marker of the cells of our interest. So this is the antibody that can attach to the cell surface protein, and thereafter we have this PCR handle and the barcode, antibody barcode, which is specific to the antibodies, and then this poly A tail to stabilize this structure of antibody derived tag. So first, this antibody derived tag will bind to the cells, and it will only bind to those cells where the cell surface protein markers, where the cell surface protein marker against these antibodies are present. So after the cells are bind with the antibody, they will then go into this single cell droplet encapsulation. So what happens in this case, from one side these cells will flow, and from other sides the beads will flow, and their objective was to has this each droplet should has one bead, and also this one tagged cells that can be seen here, this microbead, this droplet that is shown here in the enlarged size, which contains the one cells and one microbead. Thereafter this cell lysis will occur within the droplet, or will be carried out within the droplet, and it will has both the barcode and at the same time the mRNA that is derived from the single cells. And thereafter, with these beads will help in the hybridization of this mRNA with the oligos, and also with the barcode of which is specific to these cells. And thereafter, after this step, we will have both all the mRNAs that are belongs to single cells barcoded with the barcoded with this antibody barcode tag, to which this antibody has been bind to the cell surface that was present before the cell was lysed. So after this step, now the mRNAs are tagged with the barcode which are specific to that cell that cell only. And thereafter, what we will do is this PCR amplification will be carried out, so that PCR amplification and of this reverse with the help of reverse transcriptase, so that they will have the DNA sequence of this mRNA, or complementary DNA sequence of this mRNA of different size. Usually it is around greater than 300 base pair. Then after that, this DNA sequence will be further amplified independently, and followed by this NGS sequencing or any other sequencing with the help of this any other single cell sequencing. We will be able to get the information about the transcriptome levels of the single cell within the single cells. So this co-indexing and transcriptome epitopes will provide the single cell mRNA changes in the single cell mRNAs, or changes in the single cell, instead of providing the information about the average change within the group of cells. So this technique is much more accurate than the previous technique, which focuses on analyzing the pool of cells, whereas this technique focuses on analyzing the single cells, changes within the single cells. So I hope you have understood how, in brief, how this co-indexing and transcriptome epitope works. If you need to understand about this co-indexing and transcriptome epitopes, you can go through the reference paper, or for the newcomer, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.